Cypherderis in the supplementary file. Cypherderis with two R's. We have a mismatch in the spelling. So as you can see, I have just recreated a Newick string corresponding to the topology. I've done this manually. Now this doesn't take too long if the tree is small and you have some supplementary data with the taxonomic names. Now this is an intake Newick review. Technically the authors have made the data available. It's here. This is a rather older one, but it's still worth a quick look. Still some lessons here. This is from last century. But the data is on Dryad and is a single file. This is common in the olden days. You get a Nexus format, and you can get a lot of different data types in here. Now, so this is quite a complex format. And so you have so here's the sequences for different genes. I don't know what this is. Character sets, some various assignments to various classes the authors have put to the sites, and there's a bunch of trees. We have 5 here, some commands. It's got some more trees. It says ML trees discussed in the text, additional constraint trees corresponding to a table. Now the problem is when it comes to synthesis, you want to incorporate this. Firstly, how do you actually choose? But it's unclear from the file which of these is which. Now, find a few of these trees, for instance. So here's some trees here. It says several trees discussed in the text, but it doesn't actually say which one. So here's a label for the tree, but it doesn't. It'd be useful if it gave a figure number. It would be one thing, but it's some codes. So if I search for this code is not even used, and then this string is the same. So it's unclear which tree this is. And again, the string doesn't match to anything in there, and it's got no figure number. So this is it's ambiguous. So the lesson here is to use more direct labeling. You could at least put the preferred tree somewhere. I think this should be used, but also you could directly match a file name to a figure would be useful. Now, a second issue is you can see the trees. You can see they've just used genus names in the trees, and similarly in the figure. However, they've actually used high resolution in the data. They've got. You can see the data matrix where it gives the gene names, the accessions for each gene fragment. You can see they've used genus and species. So there's been a lot of that's a removal of information content or loss of resolution. So for these two reasons, I just decided to manually recreate the Newick string. That's what I've done here. And this is now done. So delete everything before it, delete everything after it, add a semicolon, check there's no spaces, and done. That's now a machine readable single result corresponding to one of their main results, one of their main figures, and species names on here. As I say, I remove. It's just 2 I ignored. So in this case, for example, here this is uninformative. One of these from the matrix did have a species name, the other one does not, does it? So it doesn't really tell you much to have, other than it's monophyletic. It's difficult to synthesize. And also this one I ignored, which has a family name, no genus name. Other than that, job is done. This can now be incorporated. But a couple of lessons from here. It's labeling of data and individual trees. It can facilitate their further use, and consider the taxonomic resolution. Otherwise, job done. Till next time.